Autoimmunity. The body is normally able to distinguish its own self antigens from foreign non self antigens and does not mount an immunologic attack against itself. This phenomenon is called immune tolerance. Autoimmunity is a condition in which structural or functional damage is produced by the action of immunologically competent cells or AB against self antigen. Autoimmunity literally means protection against self, but actually it implies injury to self, and therefore sometimes the term is also under criticism. And then autoimmune disease results from the activation of self-reactive T and B cells that, following stimulation by genetic or environmental triggers, cause actual tissue damage. Four factors influence the development of autoimmune disease. These factors are genetic, viral, hormonal, and physico-neuronic. And autoimmune diseases result from the activation of self-reactive T and B cells that, following stimulation by genetic or environmental triggers, cause actual self-tissue damage. So, four factors influence the development of autoimmune diseases. These factors are genetic, viral, hormonal, and physico-neuroimmunologic. The influence of stress and neurochemicals. All four of these factors can affect gene expression, which directly or indirectly interfere with important immunoregulatory actions. Based on the site of involvement and nature of lesions, autoimmune diseases may be classified as hemocytolytic, localized or organ-specific, systemic or non-specific, and transitory diseases. Important examples of autoimmune diseases in human and their respective autoantigen are given below in the table. Some autoimmune diseases in humans. Disease autoimmune hemolytic anemia, Rh blood group is the autoantigen. Then Graves' disease, thyroid stimulating hormone receptor, multiple sclerosis, myelin basic protein, myasthenia gravis, acetylcholine receptor, rheumatoid arthritis, unknown synovial joint antigen, then systemic lupus erythematosus, lupus erythematosus. DNA histone SNRMP, then type 1 diabetes mellitus, pancreatic beta cell antigen, then scleroderma, centromeric proteins in fibroblast. Transplantation. The immune system has evolved as a way of discriminating between self and non-self. This discriminating power of the immune system between self and non-self is undesirable in case of tissue transplant from one individual to another for therapeutic purposes. Indeed, result of transplants culminates in the phenomenon of graft rejection. Before this discussion about immunological mechanisms associated with graft rejection, it is important to understand the various gradations in relationship from donor to recipient. Graft between genetically identical twins, syngenic, is known as isograft or syngraft. In humans, an isograft can be performed between monozygotic twins. Transplants between genetically different individuals within a species are termed allografts. A xenograft is a graft between individuals from different species. Similarly, an autograft is a graft of transplant from one area to another on a same individual. So, transplanting tissue that is not immunologically privileged generates the responsibility that the recipient cell will recognize the donor's tissue as foreign. This triggers the recipient's immune mechanism, which may destroy the donor's tissue. Such response is called graft rejection reaction. Some transplanted tissues do not stimulate an immune response. For example, a transplanted cornea is rarely rejected because lymphocytes do not circulate into the anterior chamber of the eye. This site is considered an immunologically privileged site. Another example of a privileged tissue is the heart valve. A tissue rejection reaction may occur by two different mechanisms. First, foreign class 2 MHC molecules or transplanted tissue or the graft are recognized by host T helper cells, which aid cytotoxic T cells in graft destruction. So cytotoxic T cells then recognize the graft through the foreign class 1 MHC molecule. This response is much like the activation of CTLs by virtually infected host cells. The second mechanism involves the T helper cells reacting to the graft and releasing cytokines. The cytokines stimulate macrophages to enter, accumulate within the graft, and destroy it. The MHC molecules play a dominant role in tissue rejection reaction because of their unique association with the recognition system of T cells. Then immunotechnique. Precipitation reactions. Precipitation results from the interaction of a soluble antibody with a soluble antigen to form an insoluble complex. Remember, both are soluble. That means antigen and antibody are soluble, but still the result is an insoluble complex. And antibodies that disaggregate soluble antigens are called precipitants. So formation of an EGAB lattice depends on the variety of both antibody and antigen. The antibody must be bivalent or a precipitation reaction and monovalent Fab fragments cannot form precipitate with the antigen. Similarly, the antigen must be either bivalent or polyvalent, that is, it must have at least 2 copies of the same epitope or have different epitopes that react with the different antibodies. Then precipitation reaction in fluids. Precipitation occurs maximally only when there are optimal proportions of the two reacting substance, antigen and antibody. Hence, an insoluble antibody-antigen complex formation occurs within a narrow concentration range known as the zone of equivalence. This represents the conditions under which macromolecular antigen or antibody complexes are formed that are sufficiently large to be precipitated. Outside the equivalence concentration, conditions known as antigen or antibody excess occur, which result in the formation of small soluble complexes. Precipitation reaction in fluids. Precipitation occurs maximally only when there are optimal Precipitation reaction in fluids. Precipitation occurs maximally only when there are optimal proportions of the two reacting substances, antigen and antibody. Hence, an insoluble antigen-antibody complex formation occurs within a narrow concentration range known as a zone of equivalence. This represents the condition under which macromolecular antigen or antibody complexes are formed that are sufficiently large to be precipitated. So, outside the equivalence concentrations, conditions known as antigen or antibody excess occur, which result in the formation of small soluble complexes. An increasing concentrations of antigen are added to a series of tubes that contain a constant concentration of antibodies. Variable amounts of precipitate form. If the amount of the precipitate is plotted against the amount of antigen added, a precipitant curve as shown in the figure below is obtained. So you could see this figure. The precipitant reaction, x-axis antigen concentration, y-axis amount of precipitate, zone of antibody excess, equivalent zone, zone of antigen excess, we could see. And the precipitation reactions in gels. Precipitation reactions carried out in agar gels are referred to as immunodiffusion reactions. When Ag and Ab diffuse toward one another in agar, or when Ab is incorporated into the agar and Ag diffuses into the Ab containing matrix, a visible line of precipitation will form. Visible precipitation occurs in the region of equivalence, whereas no visible participants forms in reactions of antibody excess or Ag excess. Two types of immunodiffusion reactions can be used to determine relative concentrations of antibodies or antigens, as well as the identity of antigens. These immunodiffusion techniques are radial immunodiffusion and double immunodiffusion. There is a radial immunodiffusion technique, also called Mancini method. The relative concentration of an Eg can be determined by a simple quantitative assay in which an Eg sample is placed in a well and allowed to diffuse into agar containing a suitable dilution of an antiserum. At the region of equivalence, a precipitation ring form around the well, the area which is proportional to the concentration of Eg. By comparing the area of precipitation ring with the standard curve, the concentration of the antigen sample can be determined. So here we could see AB containing agar, single radial diffusion, precipitation ring, then double immunodiffusion. In the Outerloni method, I'm not sure about the pronunciation, sorry. Both Ag and Ab diffuse readily from wells towards each other by establishing a concentration gradient. It is an effective quantitative tool for determining the relationship between Ag and number of different Ag-Ab systems present. Double diffusion will be taking place. Precipitation lines. The pattern of precipitant lines that form when two different Ag preparations are placed in adjacent wells indicate whether they share epitopes. Identity occurs when two antigens share identical epitopes. Identity occurs when two antigens are unrelated, share no common epitope. The antisera forms an independent precipitin line with each AG and the two lines cross. Then partial identity occurs when the two AGs share some epitope, but one or the other has a unique epitope. The antiserum forms a line of identity with the common epitope and the curved spur with unique epitope. Identity, non-identity, partial identity. Next, agglutination reactions. Agglutination is the visible clumping of particulate antigen when mixed with antibodies specific for the particulate antigens. Antibodies that produce such reactions are called agglutinants. Agglutination reactions are similar in principle to precipitation reactions, just as an excess of antibody inhibits precipitation reactions. Such excess can also inhibit agglutination reactions. This inhibition is called the prozone effect. When the antigen is an integral part of the surface of a cell or other insoluble particle, the agglutination reaction is referred to as direct agglutination. Okay. When the agglutination reaction takes place between antibodies and soluble antigen that has been attached to an insoluble particle, the reaction is referred to as passive agglutination. And immunoassays are based on the specific antibody-antigen reaction. All immunoassays depend on the measurement of fractional binding site occupancy of the antibody by analyte. So non-competitive immunoassays directly measure occupied sites, while competitive assays are based on the detection of total and unoccupied sites by analyte. In competitive immunoassays, labeled and unlabeled analytes are simultaneously exposed to the antibody. In non-competitive immunoassays, the labeled antibody detects the bound analyte. So maximal sensitivity will be reached by decreasing the amount of antibody in competitive assays and increasing the antibody concentration in non-competitive assays. Here the black dots represent labeled antigen and white dots represent unlabeled antigen. And here you could see the competitive immunoassay. Next, labeled antibody, non-competitive immunoassay, second picture. Then radioimmunoassay, RIA. Radioimmunoassay is a technique for determining the concentration of a particular molecule in a sample based on displacement of a radioactive antigen from an antibody. Pearson and Yalow developed the RIA method and first applied it to the assay of human and animal insulins in plasma. It is one of the most sensitive techniques for detecting AG or AB. The principle of RIA involves competitive binding of radiolabeled antigen and unlabeled antigen to a high affinity antibody. The labeled antigen is mixed with antibody at a concentration that saturates the AG-AB sites of the AB molecule, and then increasing amounts of the test sample containing unlabeled antigen of unknown concentration are added. The AB does not distinguish labeled from unlabeled antigen, and so the two kinds of antigen compete for available binding sites on the AB. And with increasing concentrations of unlabeled AG, more labeled antigen will be displayed from the binding sites. Then enzyme-linked immunosorbent assay. Enzyme immunoassays exploit an enzymatic reaction for detecting the immune reaction. In 1971, Inkwall and Perryman and Van Wieman and some other scientists described independently the use of enzyme-labeled agents. The most common type of enzyme immunoassays in use is enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay, commonly known as ELISA or EIA, is similar in principle to RIA, but depends on an enzyme rather than a radioactive label. An enzyme conjugated with an AB reacts with a colorless substrate to generate a colored reaction product. Such a substrate is called chromogenic substrate. A number of enzymes have been employed for ELISA, including alkaline phosphatase, horseradish peroxidase, urease, and beta-galactosidase. Different variants of ELISA have been developed, allowing qualitative detection or quantitative measurement of either antigen or antibody. Each type of ELISA can be used qualitatively to detect the presence of AB or AG. Alternatively, the standard curve based on known concentrations of AB or AG is prepared, from which the unknown concentrations of sample can be determined. An indirect ELISA, AB can be detected or quantitatively determined with an indirect ELISA. Serum or some other sample containing primary AB1 is added to an AB coated micro titer well and allowed to react with AG attached to the well. After any free AB is washed away, the presence of AB bound to the AG is detected by adding an enzyme conjugated secondary anti-isotype AB2 which binds to the primary AB. Any free AB2 then is washed away and the substrate for the enzyme is added. The amount of color reaction product that forms is measured by specialized spectrophotometric plate reader, which can measure the absorbance of all of the wells of a 96 well plate in less than a few minutes. Indirect ELISA is a method of choice to detect the presence of serum antibodies against HIV. Add substrates and measure color. Antigen coated well, wash, and specific antibody to be measured, wash. Add enzyme conjugated secondary antibody, wash, again add substrates and measure color. And sandwich ELISA. Antigen can be detected or measured by sandwich ELISA. In this technique, the antibody rather than the antigen is immobilized by a micro titer well. A sample containing antigen is added and allowed to react with the immunized antibody. After the well is washed, the second enzyme linked AB specific for a different epitope on the antigen is added and allowed to react with the bound AG. Any free second AB then is washed away and a substrate for the enzyme is added. The amount of colored reaction product that forms is measured. Antibody coated well, wash, add antigen to be measured, wash, add enzyme conjugated secondary antibody, again wash, add substrate and measure color. Then immunofluorescence. AB could be labeled with molecules that have the property of fluorescence. So fluorescent molecules absorb light of one wavelength, excitation, and emit light of another wavelength, emission. If AB molecules are tagged with a fluorescent dye or fluorochrome, immune complexes containing these fluorescently labeled antibodies can be detected by colored light emission when excited by light of the appropriate wavelength. AB molecule bound to antigens in cells or tissue sections can similarly be visualized. This technique is known as immunofluorescence. The most commonly used immunofluorescent dyes are fluorescein and rhodamine. Other dyes such as phoerythrin and phycoerythrin and phycobiliproteins are also used nowadays. Here, antigen coated well, wash, add specific antibody to be measured, again wash, add fluorescent dye tagged antibody, supply light, colored light emission. Then immunoelectrophoresis. Immunoelectrophoresis is a qualitative technique. It combines the process of electrophoresis as well as double immunodiffusion. So electrophoresis is used for separation of antigens on the basis of charge and double immunodiffusion for the purpose of their identification. And immunoelectrophoresis is used in clinical laboratories to detect the presence or absence of proteins in the serum. In this technique, antigen measure is placed in a well in the center of site that is coated with agar gel. The antigen mixture is then subjected to electrophoresis, which separates the various components according to their charge in the electrical field. After electrophoresis, a trough is cut along the side of the slides, and antibodies to antigen are placed in the trough. So the antibodies diffuse in the agar as do the separated antigen mixture at an optimal antigen to antibody ratio for each antigen and its corresponding antibodies. Precipitin lines up from form. So identification of antigens. Identification of antigens using gel electrophoresis. Serum is placed in a well, cut in the gel and subjected to voltage gradient, which causes the various antigens to migrate different distances through the gel depending on their charge. After electrophoresis, a trough is cut in the gel into which antibodies, whole anti-human serum are placed, adapted. So next is about vaccines. An individual may be exposed to an antigen to induce formation of antibodies, a type of immunity known as an artificial active immunity. The material used to induce artificial active immunity, the antigen or a mixture of antigens, is known as a vaccine or an immunogen, and the process of generating such an immune response is immunization. Immunization is commonly known as vaccination. The term vaccine has been derived from Latin word vaccinus, meaning from cows. So vaccines can be defined as a non-toxic or non-virulent preparation of antigenic material that can be used to induce long-term humoral as well as cell-mediated immune response against pathogens. And type of antigen used in vaccines. In principle, anything from whole organisms to small subcellular fragment can be used as antigen in vaccines. Most current vaccines in use for humans consist of whole organisms described as whole organism vaccine, which include live but attenuated organisms or killed organisms and subunit vaccines, toxoid, capsular polysaccharide, and recombinant surface antigens, and live but attenuated vaccines. These vaccines are prepared by attenuating pathogenic organisms by growing them in unfavorable conditions, which result in gene mutations, due to which organism loses progeny but retains their capacity for transient growth. Attenuated vaccines have advantages as well as disadvantages due to their capacity for transient growth. These vaccines show prolonged immunogenicity and eliminate the need for repeated boosters. But a major disadvantage of these vaccines is the associated risk of reverting back to virulent form. However, these days, genetic engineering is also used to cause site-directed mutation, causing irreversible removal of virulence genes from attenuated organisms, making them safe for use. Then inactivated, killed vaccines. Inactivated vaccines are produced by inactivation of pathogenic organisms by heat or chemical treatment so that organisms are unable to multiply in the host. Inactivation is carried out in such a way so that structure of epitopes on surface antigens remain maintained. Hence, heat treatment is generally not satisfactory since it can cause protein degradation and thus loss of some epitopes. This chemical inactivation using formaldehyde or other alkylating agents is more common. Then inactivated vaccines are effective but they are less immunogenic, so often require several boosters and normally do not adequately stimulate cell-mediated immunity or secretory IgA protection. In contrast, attenuated vaccines usually are given in a single dose and stimulate both humoral and cell-mediated immunity. Then subunit vaccines. Three general forms of subunit vaccines are in current use: toxoid, capsular polysaccharide, recombinant microbial antigens. Many exotoxins can be modified chemically so they retain their antigenicity but are no longer toxic. Such a modified exotoxin is called a toxoid. Toxoids are usually not as efficient as the original exotoxin for producing immunity, but they can be given safely and in high doses. Then DNA vaccines. DNA vaccines, also known as genetic vaccines, use the genetic material of the pathogen itself to immunize the individual. In this class of vaccine fragments of pathogens genome encoding antigenic proteins are injected directly into the host cell, where they can integrate into the chromosomal DNA or exist as episomes. So, expression of genes within the host generate foreign proteins to which the host immune system responds. Hence, DNA vaccine is really an immune response made against the protein encoded by the vaccine DNA. The DNA itself is not immunogenic. Then, recombinant vector vaccines. In recombinant vector vaccines, genes that encode antigens associated from pathogen can be inserted into non-virulent viruses or bacteria. Such recombinant microorganisms serve as vectors, replicating within the host and expressing the gene product of the pathogen-encoded antigenic proteins. The antigens elicit humoral immunity as well as cellular immunity.